Kosher supervision for biotechnology labs is less an impossible new frontier than a chance to build a model of supervision that is more precise, more scientific, and ultimately more reassuring than almost any traditional food inspection. Precision fermentation and related biotech processes are driven by documentation, traceability, and sterilization. Those are exactly the strengths a Mashgichim needs. Rather than trying to retrofit old dairy check methods onto a lab, the Mashgichim's role should be reimagined as a technical partner who verifies genetic inputs, validates ingredient provenance, and monitors the fermentation life cycle the way a quality engineer would, only with an added layer of halakhic sensitivity and communal communication. Start with who the Mashgichim is and what training they need. A modern Mashgichim for biotech labs should combine halakhic competence with a baseline of laboratory literacy. That does not mean every supervisor must be a molecular biologist, but they should understand common terms: strain, plasmid, media, CIP/SIP, sterile filter, downstream purification, be able to read certificates of analysis, and know how to interpret a batch record. Training programs, run collaboratively by kashrut agencies, universities, and industry partners, can teach Mashgichim to read lab SOPs, observe aseptic technique, evaluate sterilization logs, and recognize red flags such as undocumented ingredient substitutions. Importantly, the Mashgichim's halakhic authority remains primary. Their scientific literacy simply allows them to supervise effectively and ask the right questions. Verification of genetic inputs is one of the first priorities. Before production begins, the Mashgichim should review supplier documentation for every biological strain or plasmid used: origin, lineage, manufacturer certificate, and chain of custody. Where possible, the certifier should require a signed declaration from the strain supplier confirming no animal origin materials were used in strain creation or maintenance. For additional assurance, routine genetic identity checks, PCR assays, or short sequence verification can match the received strain to a known genetic fingerprint on file. These assays are standard in biotech and can be run by an accredited third-party lab. The Mashgichim should review and archive results. When new or proprietary strains are introduced, a one-time deeper sequencing or independent validation provides confidence that nothing derived from non-kosher animal tissue is hidden in the lineage. Growth media and feedstock inputs demand equal scrutiny. Every carbon source, nitrogen supplement, vitamin, or peptone must have provenance documentation. The Mashgichim should verify supplier kosher certificates, certificates of analysis, and bills of lading. Where suppliers cannot provide clear information, require substitution with verified, synthetic, or plant-derived alternatives. Batch-level certificates, COAs, should be linked to production runs so that every fermenter run has a documented ingredient bill tied to a batch number. Digital systems make this easy. Require scanned COAs attached to the electronic batch record before the run can begin. Facility controls are a major halakhic advantage in biotech. They are also an area for focused Mashgichim attention. Bioreactors are closed systems, and CIP/SIP cycles are powerful tools for ensuring no prior non-kosher residue remains. Mashgichim should review CIP/SIP protocols, verify sterilization temperatures and hold times, and inspect sterilization logs. Periodic swab tests, ADP or microbiological, on critical surfaces confirm cleanliness. If equipment is shared with non-kosher runs, require validated cleaning and requalification steps before kosher production. Where feasible, demand temporal segregation, kosher runs scheduled after validated sterilization, or physical segregation, dedicated lines or dedicated modular skids, to minimize risk. Downstream processing is where invisible contamination risk can creep in. Filters, membranes, resins, solvent residues, and carriers must be checked. Mashgichim should review material safety data, supplier origin, and whether any consumable is animal-derived, e.g., gelatin filters, certain chromatography resins. If a consumable is questionable, either replace it with an acceptable alternative or require evidence that it is removed completely and will not leave halakhically significant residue in the final product. Retain retention samples of each lot and require independent lab testing for residual proteins or animal markers when necessary. Digital supervision transforms oversight. Encourage facilities to maintain immutable electronic batch records, EBRs, with time-stamped approvals, ingredient attachments, sensor logs, pH, dew, temperature, and CIP/SIP histories. Mashgichim should have secure read access to logs and a real-time dashboard showing active runs and alarms. For deeper assurance, some certifiers will require hashed records or blockchain-style custody seals for critical steps, so an audit cannot be retroactively altered. CCTV coverage of critical rooms, with appropriate privacy safeguards, lets a Mashgiach review handling and entry protocols remotely. Many modern Mashgichim alternate on-site presence with monitored remote supervision backed by automated alerts. Sampling, testing, and retention policies are practical keystones. Require pre-run and post-run sampling for microbial contamination, allergen testing if relevant, and residual animal markers. Keep retention samples for a defined period, e.g., 1 to 2 years, tied to lot numbers, and maintain a chain of custody log for every sample. If a consumer or rabbinic body raises a question, these samples permit independent testing. For high-risk runs, set requirements for third-party laboratory verification of the absence of animal proteins or non-kosher DNA markers. Communication and change control are essential halakhic mechanisms. Any change, new strain, new supplier, new buffer, repackaging, must trigger a change control form reviewed by the Mashgiach and the certifier. Production should not proceed without documented sign-off. This proactive model prevents surprises and aligns with halakhic concerns about unknowable admixture. Likewise, certifiers should publish clear policies on acceptable inputs, preferred suppliers, and the documentation they require, so manufacturers can design compliance into their workflows from day one. Pesach, allergy, and Marit Ayin considerations should be integrated into standard protocols. For Pesach, require vendor statements about chamet's origins of sugars or processing aids, and be ready to perform pre-Pesach inventories or certify dedicated lines. For allergen safety, ensure labels and COAs reflect allergen testing. The Mashgiach's role here is to coordinate with the manufacturer so that kosher and allergen transparency go hand in hand. For Marit Ayin and consumer perception, require front-of-pack language that clarifies ritual status and any allergen risk, and encourage manufacturers to provide a certifier-approved explanatory page linked by QR code. Finally, build a collaborative ecosystem rather than an adversarial one. Labs that view Mashgichim as partners, helping to design kosher-by-design production flows, advising on supplier choices, and co-creating SOPs, will find certification faster and more robust. Certifiers should develop technical advisories, template SOPs, acceptable material lists, and an accreditation track for kosher biotech-ready facilities. Periodic joint audits with scientific advisors, clear escalation pathways for unusual findings, and public-facing transparency, e.g., certifier statements, QR-linked documentation, will create consumer trust and market uptake. The result is a supervision model that is rigorous, modern, and deeply positive. Mashgichim become both guardians of halacha and technical stewards of safety and traceability. With training, digital tools, scientific partnerships, and clear protocols, kosher supervision of biotechnology labs can be more exacting and more reassuring than any prior food system, turning a potential halakhic headache into a model for how tradition can partner with science to serve the community.